Very good morning to you all. Today we will discuss the structure of tRNA or transfer RNA. This is also known as the adapter molecule. Robert Holly in the year 1965 reported the first base sequence of a tRNA molecule. The tRNAs are small RNAs which are having generally 73 to 90 bases long. It looks like they are having base pairs, but there is no base pair. They have some complementary bases, so they looks like double-stranded RNA. Three-dimensionally, the structure of tRNA looks like a L-shaped molecule, but when you decipher the proper structure of tRNA, it looks like a clover leaf structure. tRNA is having 4 arms. The first arm is amino acid acceptor arm. This is named as amino acid acceptor arm because the amino acid actually join to this arm. You can see here an amino acid is joined to this arm. So generally this arm, this end is known as amino acid acceptor arm. Next arm is T-Psi-C arm. Sometimes it is also known as T arm. This arm is named because they have the presence of this triplet sequence, that is T, Psi and C. The Psi is actually representing pseudouridine. It's a modified base of uracil. Next arm is the anticodon arm. The anticodon arm is the arm which contains the triplet of nucleotide, complementary bases that of codon. You know, mRNA is having codons. These codons are actually recognized by tRNA, and those tRNA which are recognizing these codons are actually bringing the amino acid to the site of protein synthesis. So here we can see the anticodon arm is wearing the triplet which is representing complementary bases of that codon. That means genetic code. You have seen 64 code, and actually all these codes are represented by different amino acids, and these codons are actually recognized by anticodon arm of tRNA. The next arm is D arm. The arm is named because of the presence of a special base. Again, it's a special uracil dihydrouridine in a modified base of uracil. So we can see pseudouridine, a special base, a dihydrouridine. These are some special bases which are only seen in tRNA. There is an extra arm. The function of this arm is still not exactly discovered. They may have variable in length in different organisms. Now, depending on the interaction between codon in mRNA and anticodon in tRNA, this is anticodon and this is codon, their interaction actually help in protein synthesis. The tRNA molecule usually accepts only one specific amino acid. Generally tRNA accepts only one specific. That amino acid is depending upon the anticodon, and this anticodon recognize codon. So we can say based on this codon there will be an anticodon, and this anticodon on the anticodon arm actually decides the amino acid that is to be added to the amino acid acceptor arm. Since there are 61 codons, you know, among 64 codons, if you minus the 3 stop codons, there are 61 codons, and each representing an amino acid. However, the number of tRNA molecules in a cell That means types of tRNA. It is less than 61. It is always less than 61. So we will have a confusion. 61 codons to be recognized by 61 tRNAs, but there are only less number of tRNAs. That means one tRNA can recognize more than one codons. How is this possible? Because one tRNA molecule can recognize more than one codon. How they are doing it? The answer is that it does so by Wobble base pairing. There is a special base pairing, that is Wobble base pairing. By this Wobble base pairing, one tRNA can recognize more than one codon. This means that the pairing between anticodon of the tRNA and the codon of mRNA always follows Watson and Crick base pairing. You know, G, C, A, U, G, C. This is proper Watson and Crick base pairing rule. Now, when you see the same tRNA which is recognizing the codons CUC as well as CUU, both these codons represent same amino acid, that is leucine. So same RNA, same tRNA can recognize these two codons. Let us see how. One is by normal base pairing, G to C, A to U and G to C, no doubt. But if this codon comes to the site of protein synthesis, the same tRNA can recognize that also. How? G to C, first codon or first nucleotide of the codon 1 from the 5' end. Second, AU strict base pairing, AB Watson and Crick hai, AB Watson and Crick rule hai. But when you go to the third base, you can see it does not follow Watson and Crick pairing. There is a wobbling occurs in the third base. Here, even though it is U, that is recognized by the guanine, there is a base pairing occurs between G and U. This base pairing is known as Wobble base pairing. Wobbling occurs because of the conformation of the tRNA anticodon allows flexibility at the first base of anticodon. You can say this is first base of anticodon or third base of codon. We say always from 5' to 3'. First base of anticodon, yeah, third base of codon. So all the codons that differ in the third position and code for same amino acid can use a single tRNA in protein synthesis. Thus we can decrease the amount of tRNA in the cell. That means one tRNA can recognize all the same amino acid representing codons. Okay, that's all regarding tRNA. Charging of tRNA means binding of amino acid to the amino acid acceptile arm. This binding is catalyzed by an enzyme, that is amino acid tRNA synthetase. These group of enzymes, they are actually controlling or catalyzing the process of charging of tRNA. See, the overall reaction is amino acid plus tRNA plus ATP, it requires energy, forms amino acyl tRNA plus AMP and inorganic phosphate. So this is one of the rarest reactions in the cell, where an ATP is converted to AMP. 3-phosphate group ATP is converted to 1-phosphate group, that is AMP. It releases huge amount of energy. This happens only at certain times. In majority of the reaction, what happens in our cell? ATP is converted to ADP. That means 3-phosphate will become 2-phosphate. It itself releases energy, but some rare reactions requires high energy, so there ATP is converted to AMP. Charging of tRNA is such a reaction requires huge amount of energy. So based on the anticodon, the amino acid is charged at the amino acid acceptor arm. This is acceptor stem and this is acceptor end. There the amino acid is added by an enzyme amino acyl tRNA synthase. Okay, that's all for today. Thank you.